Good morning everyone. We the students of Central University of Kerala currently pursuing our masters in botany from Department of Plant Science. So today we are here with the research article analysis on the topic Autophagy-mediated degradation of integumentary tapetum is critical for embryo pattern formation, which was published in the Nature Communication, and the authors of this article are Lin-lin Zhao and et al. So I am Agilvi and my team members are Bhavya K and Nihara Vijay.
We are delighted to delve into the fascinating realm of plant biology, particularly focusing on a crucial yet often overlooked process in seed development, autophagy. Autophagy, a cellular mechanism responsible for the degradation and recycling of cellular materials, has emerged as a central player in various facets of plant growth, development, and stress responses. While its significance in male gametophyte development has been well documented, its contribution to seed development remains a tantalizing frontier awaiting further exploration.
This work aims to explore the role of autophagy in seed development, specifically in relation to its role in the degradation of the integumentary tapetum and its effects on seed viability and embryo pattern creation.
The major objectives of this study have to examine the functions of autophagy in the initial stages of seed formation with a specific emphasis on the integumentary tapetum's breakdown. Autophagy can be classified into three main categories such as chaperone-mediated autophagy, macro-autophagy and micro-autophagy. A set of conserved core autophagy genes, which is popularly known as ATG genes, which are grouped into functional groups during autophagosome formation, tightly regulated autophagy. The five functional groups that these core ATG proteins belongs to during autophagosome formations are the phosphatidyl inositol 3 kinase, which is known as PI3K complex, the ATG8 and ATG12 ubiquitin-like conjugation system, the ATG9 cycling system, and the ATG1 to ATG13 complex. These are some of the examples of the ATG genes. So we have to evaluate the consequence of inhibiting autophagy by mutations in the autophagy specific genes ATG5 and ATG7 on the development of the embryo and the breakdown of the integumentary tapetum. So we have to undertake a comparative lipidomic investigation to clarify the functions of autophagy in lipid metabolism at various phases of the seed germination. Programmed cell death, which is popularly known as PCD, causes and the tissue to degenerate. This process is essential for the development of the pollen wall and male fertility.
Experiments were conducted on Nicotiana tabacum, a major plant requiring short-day conditions. For molecular level studies, the tobacco genomic DNA was isolated and amplified, and ovules and seeds from wild type and autophagy-related gene mutants were collected. The methods employed for this study include vector construction and plant transformation techniques, immunofluorescence assay for achieving a high resolution image of the sections, tunnel assay, body pie staining, embryo isolation and mRNA extraction, RNA sequence library construction, immunology assay, and vascular processing enzyme activity assay for understanding programmed cell death.
The constructs pro-ATG5H2B-GFP and pro-ATG7H2B-GFP were generated using the CRISPR-Cas9 genome editing technology, and PCR technology is utilized for the introduction of target sequences into corresponding single-guide RNA expression cassettes. The immunofluorescence assay was conducted on the sections obtained from seeds and ovules of wild type and ATG mutants according to the assay protocols. Phosphate buffered saline and isotonic solution was used for washing for multiple times, and incubation was done using anti-ATG8A and anti-ATG7 antibodies. The sample observation done under confocal laser scanning microscope with the excitation at 488 nanometer and 561 nanometer. This is a diagram elucidating how the mutants were generated in order to showcase the role of autophagy-related genes mutation in blocking the autophagosome using CRISPR-Cas9 system. In diagram, blue box is the exon, grey line is the intron, and pink depicts untranslated region. The schematic diagrams of B shows the domains within the ATG5 and ATG7 proteins. Missense sequences are depicted as green box.
This is the image of terminal deoxynucleotidyl transferase DUTP-NIC-N labeling assay, which is popularly known as tunnel assay, is used for detecting DNA fragmentation in which the paraffin sections were dewaxed with the silane and rehydrated with alcohol, fixed in 4% paraformaldehyde for 5 minutes as per the protocol. The final reaction was terminated in saline sodium citrate solution for 15 minutes, and sample were washed by the fresh PBS. It is observed under the microscope under the excitation 488. The immunoblast assay characterized ATG protein level and the extract were later subjected to SDS page and subsequently transferred to nitrocellulose membrane and immunoblotted with the anti-ATGS antibodies.
The functions of extra-expressed genome is analyzed through transcriptomic study, and hence, in this study, 8 cell embryos were collected for mRNA extraction and library construction. The raw reads obtained through the RNA sequence data analysis were filtered using FAST-P and eliminated low-quality reads and adapters, and then aligned to Nicotiana tabacum plant genome. The read counts and FPKMS were calculated using RSEM RNA sequence by expectation maximization.
Data analysis. Since the study particularly analyzes the autophagy phenomenon at molecular level, several methods of study were implemented. Hence, different data analysis tools are incorporated into this, such as image analysis, lipidome analysis for lipidome profiling, and statistical analysis for the incorporation of obtained results, the immunofluorescence assay, tunnel assay, and body pie staining, which is fluorinated boron dipyromethane staining. These samples were analyzed through confocal laser scanning microscope. In lipidome analysis experiment, from the mixture of test samples, only 4 samples are used as quality control sample for the analysis. The quantification was done using triple quadrupole mass spectrometer.
So we moving on to the result. During seed development, autophagy is active in the integumentary tapetum as evidenced by various analyses. Firstly, the use of reporter lines expressing GFB, green fluorescent protein, fused to ATG5 and ATG7 revealed their expression within the integumentary tapetum. This finding was further supported by immunofluorescence labeled on autophagosomes using ATG8 antibodies, which displaced in the tapetal cells. The confirmation of typical autophagosomes and autophagic bodies in the integumentary tapetum through transmission electron microscope provided additional validation. These experiments were repeated multiple times in the constituent result, confirming the activity role of autophagy in the integumentary tapetum during the seed development.
The ATG5 and ATG7 mutants generated using CRISPR-Cas9 exhibited reduced protein levels compared to the wild-type plants. Immunofluorescence imaging showed a fewer autophagosomes in the integumentary tapetum of mutant plants. Additionally, the levels of ATG8-PE, essential for the autophagosome formation, were decreased in the mutant lines. Furthermore, the mutants displayed a higher frequency of seed abortion compared to the wild-type plants. This defect was skewed by the introduction of a transgene expressing ATG5-GFP. These results highlight the crucial role of autophagy in seed development. Upon careful analysis, abnormalities in the integumentary tapetum and embryo development were observed in ATG5 and ATG7 mutant seeds. Specifically, the integumentary tapetum in the mutant seeds exhibited excessive expansion and delayed degradation, which was not observed in the wild-type seeds. This defect was fully restored by introducing a transgene expressing ATG5-GFP. Similarly, abnormal embryo pattern formation was detected in the mutant seeds, with a higher percentage of embryo displaying irregularities in both apical and basal cell lineages compared to the wild-type seeds. These findings highlight the critical role of autophagy in proper development of endothelium and embryos during seed formation. Using techniques like tunnel staining and transmission electron microscopic analysis, it was observed that in wild-type plants, programmed cell death starts around 4 days after pollination and is fully established by 5 days after pollination. But in autophagy deficient mutants, PCD initiation is delayed with fewer cells showing DNA fragmentation and reduced tunnel signals during seed development. This suggests that autophagy acts upstream of PPE in regulating PCD pathway in integumentary tapetum. Overall, these findings highlight the essential role of autophagy in initiating timely PCD in the integumentary tapetum during seed development.
So we moving on to the discussion. Autophagy, the process of cellular self-degradation, is crucial for programmed cell death, particularly in facilitating the degradation of the tapetum during the seed development. Similar to the anther tapetum, the integumentary tapetum also undergoes PCD, playing a vital role in the shaping the embryo pattern and ensuring proper seed development. This particular research has revealed that the integumentary tapetum communicates with the embryo, which is essential for ensuring proper embryo development. This was evidenced by the experiment involving ATG5 expressing rescue, which demonstrated the importance of this communication. To summarize, this research article undergoes the critical role of autophagy in coordinating the timely programmed cell death of the integumentary tapetum, thereby ensuring the proper development of embryonic patterns and early seed formation. This research highlights the significance of autophagy as a fundamental mechanism in regulating key processes essential for seed development and lays the groundwork for the exploration into its intricate molecular mechanism and potential applications in agriculture and biotechnological context. So we conclude our analysis. Thanks for patiently listening. If you have any doubts, suggestions or any questions, kindly please text us in the chat box. We will surely respond. So once again, thank you so much.